Okay. So I would like to start here for session 5. Session 5 is advanced technology and characterization of complex drug products. So this is kind of getting into some really cool new kind of novel techniques and modeling stuff. So be prepared for that. First up, we have Xiuling Liu, from University of Connecticut. And she's going to be sharing imaging in situ forming implants using advanced characterization. Please welcome.
Thank you for the introduction. Good morning, good afternoon, good evening for everyone online. So today I will share some of our research, but before that, from our very active discussion session before and all the sessions before, we can tell there's a need of further understanding. So like there are variations from polymer, PLGA, molecular weight, to blockness, and all the properties. There's tons of variations in terms of batches and in terms of lots, right? So in terms of the implant that we studied, then in order to get some regulatory flexibility, I would say we need to be confident about our understanding. If we understand what are real critical quality attributes, we understand the flexibility of that range we can get leading to the in vivo performance, then we could, that's my understanding, we could look into the case-by-case flexibility. Is all there. So then coming back, we would like to present this, some of our work recently we developed to try to understand better for the in situ forming implants, and appreciate the support from FDA. And then we work with our colleagues at University of North Carolina, Chapel Hill Small Animal Imaging Core facility for the image analysis. So we want to know more about what happens in the implant itself once it contact aqueous, either in biological environment or even in vitro. We want to know the structure of that and how that will correlate to the release of a drug. So we start with our model drug comp product. It is Eligard. So we all know that it has two parts. One syringe contains a drug, is lyophilized powder of lupus acetate, and the other is a syringe containing etrogel, as a mixture of PLGA and the solvent NMP. So that's a well-mixed solution. And then we can inject the sub-Q space, and then you get the water aqueous exchange with the air, with the solvent. So then you get the solidification of polymer and subsequent drug release, as well as degradation as facilitated drug release. That's a typical understanding. So to study all the in situ forming implants, we really typically use conventional method as in vitro release method, but there is no standard in vitro release method. Basically, you develop your own. And we also look into the in vivo pharmacokinetics, trying to understand in release, deconvolute to the release locally. Well, then there are variations between animals or between individuals. So that gives us lacking of understanding, kind of black box, trying to play the stats. And there are other factors could affect the performance, like the way you mix in syringe and the way you inject, and the physiological environment could also affect the drug release and the subsequent performance. So what we want to do is, first, we want to get some insights through imaging. So we started with CT contrast agent, which is called iHexo. It's approved in clinic that can be used to inject to patients for CT imaging. So we mix that together with the drug lupus acetate. And that is one part. And then the other part, no changes, still NMP as a solvent and the PLGA. Well, we started to do in vitro testing and just mixing that. And then, okay, so I have a mouse here. You can do in vitro, just inject that mixture, well, mixed into aqueous, and then we get the form of implant in aqueous, and we look at in vitro release, and we also take the implant. Basically, we took an implant and we image that. We also weigh the implant, and then we can monitor all this drug release as in the meantime. And some of the implant we took out to do SCM image. So the other study we also did in vivo, just injected implant into the subcu space of rats so that we can use imaging as a CT to look at the implant. So the next thing, I hope you can see that clearly, that we're looking at the different formulations at different time frame. Actually, we did across up to 30 days because this is meant for 30-day release of drug. And we compared 4 groups. It's interesting to see the 4 groups. Basically, first we had just HGL and there's no drug. And we also have just iHexol as an imaging agent is representing our drug as a whole. And we also have lupulin acetate as LA, which is a drug. And the 4th is the mixture of iHexol and lupulin acetate. So, well, we took the saying like first and no drug and all the formulations, you see like we can see the contrast, and you see a brighter signal that's mainly our contrast agents deposition as iHexol deposition. And interestingly, when we bring up 2 images, like 8 hours of iHexol versus iHexol plus lupulin acetate, and then you can see just the differences by appearance. And if you only have iHexol, you have first, they both showed a denser shell, it's a very thin shell that we know that you drop this solvent into aqueous, the surface of the polymer will solidify first and form that structure. So that gets a denser shell for both cases. But for the iHexol only, you can see that it has a very high intensity core, which represent iHexol is accumulated in the core. And the surface, you also can feel like a little bit deposition of iHexol is trying to escape. They're trying to, diffuse out from the core. And in terms of the mixture of lupulin acetate and iHexol, it's very different. So you see like a bulk part is a homogeneous, a mix of iHexol and, well, the matrix, if we say that. And then you have a little bit dimmer of a surface at the first place. What happened to that? So like, I would like to show you the release first, and then we go back to that. So it's interesting to see that iHexol release, that is very hydrophilic compound. It's very easy to mix with the solvent. And then if we only have iHexol, and you see that release is very fast. But when we have a drug, a lupulin acetate and iHexol mixed together, and the release tend to be very slow. And based on the lupulin acetate by itself or the mixture together with iHexol, there's no change. So then what happened is, like, if you look at the NMP release, basically NMP release curve for no drug and iHexol only is blue and red, you can see it is very consistent with iHexol release. So mainly this iHexol is very hydrophilic, and it's really hand in hand together with the NMP. Well, then when the NMP going to this mixture where LA by lupulin acetate itself, or with a mixture, the NMP release, the solvent release is drastically changed. So that release is quite slow, which you can see the consistency of hexyl and the lupyl acetate. That just shows that when you have the lupyl acetate, and that influences the polymer from the implant formation, and it gets slow down of all the drug release. I want to go back to this. So the image shows you this is a fast phase inversion process. I appreciate actually Dr. William Smith's talk about all the phase diagram and all the phase inversion process applies here, that you get very fast phase inversion and then the rapid drug release together with the NMP release gives you this layer. And then for the situation with lupyl acetate, that is giving you a very typical slow phase inversion process. So that is slowed down and then allows first the formation of a shell layer, and then you get very well distributed polymer together with lupyl acetate and hexyl, evenly quite distributed largely in the implant. So basically you have this kind of very distinct differences, and depends on the drug that it has, which impacted the release of a whole for the formation of implant and the release of the drug is subsequent. So when you are able to monitor that as a different time, you can see the size and shape changes. So then we took, just go to this slide, that we took the implant, we can monitor the weight, whole weight of the implants, and also we are able to use imaging to quantify the volume of implant. So the left figure shows the volume, and in the middle is the weight of the implants. You see that the very consistent trend of the implants, basically with just the two curves that on the bottom is blue and red, showing that's no drug, and hexyl, you see that fast, very fast phase inversion gives you volume, not much volume increase, and its drug release and the solvent release very quickly, and then you get a water influx in. But when you get to the this green curve is with a lupyl acetate together, purple, both showed very large volume increase and weight increase. So, and then going to the implant, this is molecular weight of implant. We took out implants at different time points. You can see the curve that with lupyl acetate, the degradation is slower compared to when you have no lupyl acetate. So all these shows that you have very fast, rapid phase inversion for hexyl case, and then that gives you very interesting trend correlated to this, all the NMP release. At A is the no drug, that you see that at the early phase, the volume increase and NMPs quickly released at first a burst and then quickly release, and then to a point they get to degradation predominant and then going to the weight decrease. So you see that release will slow down. So all these are showing quite consistent trend of that. I want to show you this because that gives you the interesting correlation that for no drug and hexyl only, you see that pore formation is larger, which we pick at the 7 days because early time point we really have hard time to get the implant out, and was able to quantify using the SEM. Even with the lupyl acetate, we are able to take that. The mixture we couldn't. So you can see this for lupyl acetate only. It has very small pores, smaller pores inside, which slow down the release of everything, then water intake and water diffusion as well. And for this case of no drug and Ihaxo only, you see a larger pore formation, which gets fast, rapid phase inversion, provides more of the polymer lean areas, and as a pore forms, and you get better drug release and influx of water exchange of that. So that gives you this phenomena. And then going to different days, and then you get some more degradation, and they're not as significant difference as earlier phase. Well, then we want to translate to the in vivo. What happens to in vivo? What are the differences between in vitro and in vivo? So we took implant, and that's the two comparison with Ihaxo and Ihaxo plus loop acetate. Because the CT contrast agent, we are able to see the implant, and they're similar, compared to in vitro. I hope you can see that Ihaxo has still showing the core shell structure, and versus the mixture has more of a homogeneous mix of the image of implant. And we are able to just monitor 7 days, and after that, actually the image was not as significant as you can tell. And we took out the implant. You can compare these two. That for Ihaxo, it's already down to like a very homogeneous stage, and because of slow release of Ihaxo together with loop acetate, and then you have still some signals you can see a bit better of the contrast. So just to compare the differences in terms of the trend, we see a similarity of the trend compared in vitro and in vivo. But in vivo, we can see that things happen to be a bit faster compared to in vitro, because we see this phenomenon probably not at 7 days, is a bit later, like 11 or 12 days. I want to show you this because we all of us talked about the source material and lot variation. So we did have another vendor that we started with that vendor, and unfortunately the vendor discontinued the product. So we are not able to really see the release and compare a lot further between those two vendors. But we are able to see very distinct differences between this in vivo imaging and compared to the group we saw just the slide before. And then you see that at the formation, that's all good. This is Ihaxo only. But as time going, you see a lot of kind of cavities. It's a black cavities. As time going, as 7 days, you can see quite a bit. And then it doesn't show in the other the other vendor. And then you are able to see, after 14 days, it's kind of a whole very void space in the core of the implant. And then eventually it clapped into a smaller implant, almost gone. So that shows it looks like it has even faster phase inversion and compared to the other polymer that we used before. So that's the polymer properties does impact the implant formation and the subsequent drug release as expected. Well, we are still having a lot of studies going on with the time. We can share a lot others, and more in vivo imaging, basically more quantitative analysis is ongoing. And we have, besides, excuse me, besides the CT, we have optical to really real time track the drug release in vivo in this non-invasive way without interference between individual of animals. So with that, I would like to summarize that we use this as imaging tool to understand better of the in situ forming implant in terms of drug deposition in vitro and in vivo, and then we are able to see that this core shell structure as well as the pore formation based on the SEM, and we are able to see the influences of ihexyl and lupullic acetate. When you have a more hydrophobic, rather more hydrophobic lupullic acetate, that does influence the mixability of our solvent and water, and the subsequent implant formation of the solidification of a polymer. And so with the in vitro and in vivo correlation gives us a bit more insights, and we will definitely do more into a quantitative measure of in vivo pharmacokinetics, and then convert to this release as well. And implant, the last point was implant made from different vendors of PLGA does show the differences. So that's what we observed. With that, I really appreciate all the collaborators from UNC FDA and other, and also Shawn, the DGM, and also our lab members. Thank you for your attention.
Okay, thank you, Professor Liu. Next up, we have Shawn Zhang. He is the founder and managing director of DGM. So he's going to talk to us about some image-based in silico modeling, density and porosity for bioequivalence. Really excited. Please welcome Shawn.
Thank you so much, Billy. And thank you, the host inviting us to give this opportunity. So continue from Xiuling's great talk, we will focus again on the product itself. The discussion we had in the past couple of days were either the earlier stage, the substance, or how we test them, right? The final product. I think the product itself contains a significant amount of information where worth actually to take a closer and deeper look. And we know, looking at the substance, right, very early phase, looking at the final product and test them, neither of them are actually very easy. So drug product is difficult to characterize, and particularly if we want to characterize as a black box from the outside. This is one very quick example. A lot of you probably have seen a typical PLGA product. And we are looking at the product made from two different processes. And if we're examining from the external product surfaces, in this case, the SEM surfaces, they look very similar. We can go through laser diffraction. We can do all kinds of more conventional testing. They are very comparable. But if we want to understand what process is associated with this product morphology, where the drugs are, and how actually we get to these products, right? This surface morphology is not sufficient. If we introduce additional technology to open them up, and in this particular case, we use a focused ion beam to create artifact-free cross-sections. Now we can clearly see interior to these microspheres, we have the minocycline crystalline particles in corresponding to process one, evenly distributed, uniform, while the particles in process two are more further encapsulated and buried inside the microsphere. And we have a drug-free zone, which is causing a 3-day delay of the drug release. These are the important information and begs the question, the equivalence assessment insertion point. Is that actually a start or an end? In our generic drug product development, we actually are fortunate of sitting on the answer. That's our idea. If you look at the development process, it's apologies for the oversimplification. We know there are a lot more than just 5 steps. But just in general, right, process formulation development, testing method development, and then we need to assess the equivalence internally from the sponsor standpoint, and then regulatory equivalent assessment, and all the way to the approval. The main challenge there is if all go in one shot and we get it, right? Great. But the iterations, right? So this is where we actually have to exercise a lot of experience, a lot of guesswork, and each iteration is costing money and resources. And the further away we are down the pipeline, the iterations is becoming more costly, right? So this is where continuing iterations can be very intimidating, particularly for complex dosage forms. Now remember, we do have our RLD, right? And the RLD is beyond just a piece of paper describing their spec. We have those products, right? So how about we take a deeper look at those products? So in this new workflow, we advocate equivalence by design, right? So before we actually even made anything, let's take a closer look at our RLD product. We collect a suite of three-dimensional correlative images, and that includes characterization on particle size, on porosity, and density, as well as a number of additional critical quality attributes. And that gives us a digital representation of the RLD product. And then we can use generative AI to predict what's going to happen to them, right? The particle size change from processes, porosity changes from processes, or density changes or stability or release. And then with both the original RLD image and generative AI predicted formulations of process products, then we can combine with the in silico dissolution based on these microstructure models. So this is a potentially important tool to give us proof of concept and ask what if validation questions that can guide the process development to complement the testing method development and accelerate the equivalence assessment. So today's talk, I'll focus on three of the properties that we focus on in the past few years, or actually a large suite of different characterization matrices we worked on. So let's first look at microsphere true density and how that can be a potentially discriminatory quality attributes. So this is a collaboration with the FDA and Dr. Burgess' team at UConn, where 8 different formulations were designed with different processing conditions. Right. We use X-ray based true density measurement, and we correlated the density variation within the microspheres. So that is characterizing the drug distribution. Are they more on the outside, more on the inside, from macrosphere to macrosphere and intramacrosphere, right? So we have these 8 formulations and their corresponding density variations. And from this assessment, the conclusion was all samples are Q1 and Q2 equivalent macrospheres. The release differences are most likely only due to Q3 microstructure differences. And then more uniform spherical density leads to slower release, better encapsulation. Now, with these correlation trends and these formulations that were designed to be different, right? How does it assist us in the equivalence assessment? So this is a structural equivalence zone, and this is currently still preliminary work in progress. And we are looking at this zone where these 2 formulations leads to most pertinent agreement with the release profile, which we actually published in the paper. So in these particular 2 cases, the DCM solvent requires more viscous silicon oil, and EA solvent requires less viscous silicon oil. And the release is less sensitive to stirring speed when it is above certain rotation speed. Right. So these are some of the assessment, equivalence assessment associating the microstructure, the release rate with the process conditions. So how do we exactly measure the true density? The density measurement on these microparticles, microspheres with porosity, with sub-resolution API, small particle size dispersion is not easy. Simple measurement such as volume weight, it's difficult to get accurate volume measurement when you have a macroporous material. To further complicate the approach, if we take images, right, we'll assume they might actually have the same intensity, which will allow us to convert that into density. That's also not very straightforward. Using X-ray as a non-invasive technique, we are able to collect these intensity images. But looking at two particular locations from the microsphere, right, there's no guarantee their intensity are the same, because X-ray has to penetrate through the whole bed of the powder. We actually have different X-ray history. And then from different sample to sample, that the X-ray history is also different. So in order to obtain this true density measurement, we designed interesting technique where we introduce the NIST standard material in each and every of our product scale. So in this particular case, we recommend 3 to 5 calibrants. We can use some of the natural inclusions, such as the air and the PLGA sample holder that we printed. Or we can use NIST standard material, such as PP, PET, and Teflon. And then once we use this standard density material, we can build a density to intensity correlation. And using this correlation function, that will allow us to convert each and every pixel in our 3D-dimensional space from intensity to its true density. So before we claim victory, we also have another issue we need to take care of. So that is this halo. This is actually a fairly notorious tomography penetration-based imaging artifact called Fresnel diffraction. So this is caused when a beam or a wave penetrating from a lower density material to a higher density material can diffracted by the interface. So in this case, we introduced the deconvolution method to deconvolute out those pixels that got contaminated by the diffraction. So this all sounds good, but does it work? We conducted a careful validation study using 2 different X-ray sources, laboratory X-ray source that is available in a lot of our labs, and then the synchrotron X-ray source. And these were also conducted at 2 different resolutions, 1.25 micron and 0.34 micron, nano CT. We introduced a particle system based on PMMA material, another NIST standard material with known density. And then we pretend we do not know them, design the experiment with all these calibrants. And then we were able to back calculate the density distribution corresponding to these particles. So with the laboratory source, we calculated the density profile with the average density of 1.221, corresponding to 1.19 of the true material. And the synchrotron data shows very similar results. So jump to another case study very quickly. Once we reconstructed this 3-dimensional microstructure data, in particular, the drug distribution and drug concentration, there are actually a lot more we can do than just looking at them or quantifying them. These in silico dissolution models were built and actually thoroughly validated on several of our programs. And this is one quick example, also extensively published with the Genetech team on ocular implant program. So this implant program intended to address the wet age-related macular degeneration. And the therapeutic approach is trying to replace this periodic injection with a 6-month long-acting PLGA-based implant system. Right? So the target duration is minimally 4 months, and then target release has to be greater than 0.8 micrograms per day. And we have other implant size, target degradation time, and of course, very important safety considerations. it has been discussed several times. Testing these each iteration will take 4 to 6 months, right? So with this imaging-based approach, we conducted in parallel the imaging, AI, generative AI, and in silico prediction, along with the Genetech team in vitro DOE. And we evaluated 8 laboratory batches, 7 GMP batches. And these are fairly non-trivial DOE space with different facility, 2 different polymer Lg ratio, 5 different drug loadings, 2 different extrusion temperatures, and 2 kinds of API with different counter ions. And these were evaluated in about 12 to 24 months. To take a quick peek at some of our in silico dissolution model, this is a blind model that we were able to build just by looking at the implant data and the parameter space without any testing data. Right? We can see this correlation with our in silico model. Here we are plotting the Higuchi rate constant between our prediction and the in vitro release profile. And the first 7 days, we have very strong correlation. In addition to the rank ordering, also very strong correlation. And this is when the polymer degradation for this 4 months release did not fully kick in. And it's primarily the drug release properties, drug solubility and diffusion properties are dominating the release. While our initial blind prediction did not do a great job throughout the entire release in late stage. Right? This is where the polymer degradation is becoming very important. And through a polymer correction step, we were able to improve our model and get much better prediction. So I'm skipping a lot of data and details, which I'll be happy to share post-slide. How about the correlation over time? Right. So this is another implant program under Gates Foundation, where we are looking at the implants at 4 different stages: day 0, 28, 56, and 84 days. Again, a lot of data here, but one interesting point I want to highlight is 56-day release. Right? This corresponds to an AI-assisted reconstruction of the microstructure features in this implant. Right? So in this implant, we're looking at the grayscale, some morphological features. These are all fairly qualitative. This is our path to quantitative and predictive. In this particular AI system, we were able to differentiate and recognize the remaining API in green, polymer in blue. And then we have 3 porosity systems. We have the pre-existing pores, this larger void space, which we also see from day 0. And then we have the evacuated pores on the outer layer. And then we also have a fracture network. Okay. The recognition of these different morphologies of pores, and more importantly, their important roles in drug release is crucial. And there is actually a lively debate in my LinkedIn recently about porosity recognition and porosity classification. That got me to 4,000 followers like overnight. I don't know it's a good thing or not yet. Coming back to this particular example, the recognition of fracture, right, immediately supported the InnoCore and the Gates Foundation team to understand why in vivo environment, there is actually a sudden intermediate burst, or it's almost like a second burst during the release, which wasn't anticipated. Right, so without actually direct imaging and characterization of these fractures, it will be very difficult to understand what exactly happened. Also, in vivo and in vitro correlation. So this is another snapshot where we were looking at 2 different in vitro states. We are looking at actually the same day, right? Same day, 56 day, but in vitro in comparison with the in vivo. We can see they are not that correlated. Right? They don't look very similar, very comparable. But if we actually look at the in vitro implant release of 28 days, we actually have a much better correlation with the in vivo 56 days. So this is going beyond just looking at 2 data points from our release curves and understand exactly how these 2 implants evolve and get to this point. So coming back to porosity measurement, this is another great collaboration with Dr. Burgess' team, where we are comparing the porosity characterization from these microspheres with a more conventional method, in this case, mercury porosimetry. Right? This is cross correlation, and we can see the conventional mercury porosimetry is fairly consistently 2 times higher than our image-based measurement. Right? And these are corresponding to 4 of the earlier formulations we saw. And mercury has almost like always a 45 to 50% porosity. Right? It's not super discriminatory. So we took a closer look at the corresponding images. So this is a intraparticle imaging, looking at the microporosity network inside the microsphere. And then this is a microsphere powder bed imaging. Right? And then we correlate the scales we observe with the mercury intrusion porosimetry data. So this is a very typical reading by pushing higher and higher pressure, right? We're getting into smaller and smaller pores and measure how much volume of mercury getting penetrated. So we can observe actually 3 very interesting zones, right? We look at this red zone, porosity in the size of a few microns to hundreds of microns. Okay. Our microsphere is only about 50 microns. We can't have 50 micron pore in a 50 micron microsphere, right? So these are actually corresponding to the intraparticle pore space, right? Which actually has very little to do with our actual drug performance. We also look at the final leg of this porosity measurement. That's a few nanometers, right? So these are the pores that are likely associated with the intrapolymer space, right? Nanometer size pores, molecular space. So that's probably also not very interesting for our purpose. And then these are actually the porosity of interests, but mercury did not have any meaningful reading. So MICP porosity measurement on soft material can be misleading. And these are actually some additional study. I may not have time to dive into it. So we lost our visual. Okay. I have to say, Dr. Burgess and Ray Feng, our collaborator, these mercury measurement, it's really chaotic that they are able to do it and get actually this measurement done. This was not easy. Thank you. Yeah, a lot of data on the right, it actually showed our image-based mercury porosimetry model. So it's still mercury porosimetry, but performed in silico using the 3-dimensional pore network. So that's this curve. And then with imaging data, we can not only characterize the pore network and the so-called pore throats, how they are connected, but also each individual pore body, which is this curve, right? Pore throats and pore body. And we can also use our X-ray microscopy data, macro CT data, to characterize the inter-microsphere space in a similar manner. Now that essentially confirmed my earlier qualitative assessment, right? So these XRM data has pore throat almost exactly as a mercury porosimetry produced. And we have the corresponding pore body. While the real meaningful and release controlling critical quality attributes porosity are actually in this range. Now with that, let me just do a quick recap. From structural equivalence by testing, as after the drug products were made to a structure equivalence by design. Right? So we have this equivalence assessment pipeline. All of us are on the same page regarding the complexity and challenges. And this summarizes some of the key points that we were trying to address with our clients and our collaborators. And why existing Q3 methodology often are deficient, right? Yeah, we summarized the particle size, the outside characterization. And I think an earlier question asking about is there a method to be truly non-invasive, right? So that method is X-ray. X-ray can be truly non-invasive. Is it applicable to each and every product? So that's a different discussion. And then the new tools. So I highlighted 3 of them today. The density measurement, very high throughput and cost effective. Right? We're measuring many thousands of microsphere particles non-invasively, right? Getting a statistically meaningful representation. And those microsphere particles can be actually sent back to you guys, to our team to test them. Right? So because it's fully non-invasive. And then in silico dissolution, we demonstrated the sensitivity study. And then we also have an image-based intra-particle characterization, which is the highest sensitivity, but it may not be also the quickest. So let me conclude equivalent characterization protocol. I won't have time to cover this animation demonstrating the entire workflow, but the primary take home is it might be worthwhile to consider spending a bit more time looking at our R&D product, right? Understanding them qualitatively with imaging, often using a correlative, 3-dimensional, and non-invasive approach. And then start build that understanding with artificial intelligence, with quantitative approaches. And then also start to ask what if questions and sensitivity questions with the in silico prediction models. So with that, thank you all very much.
Okay, next up, we're going to look at some high-resolution chemical imaging for both intrauterine systems and products containing nanomaterials. I think he's around here somewhere. He's a fellow FDA research scientist. Please welcome to the stage Huzeyfe Yilmaz.
Great. Thank you for the introduction. I am really conveniently placed in this schedule. I really appreciate that. It's really makes my job a lot easier introducing what I'm going to talk about. I am part of OPQ Pharmaceutical Quality. Move on, and so I'm going to just jump right in. And talk about a little bit about image-based characterization, what was discussed so far. When light interacts with a specimen, couple of things happen. It can be absorbed, it can be scattered, can be diffracted. Some amplitude changes happen or some phase changes happen, and it's essentially a parameter like refractive index, birefringence, but it's a univariate parameter that is used to generate contrast. I have an optical image here from a cross-section of an intrauterine system. The core part with the micronized API is dispersed in the PDMS matrix and the outer PDMS. The contrast is generated by light scattering. So it's a singular parameter. The same with the X-ray tomography or micro CT. Essentially, it's the material density that's generating the contrast. This is an long-acting injectable that's showing the voids inside the sample. That's it's the material density. It relies on that singular parameter. Not very different for electron microscopy, even though there are different types of electrons that can be generated when a beam interacts with a specimen. But at the end of the day, it's the electron density or the atomic number that determines the contrast that's generated. So the image here is actually from a sunscreen topical formulation that has multiple UV filters dispersed in a polymer matrix, we can't really generate contrast. We can see the globules of the UV filters, but we can't really generate contrast between those UV filters to really identify them, right? So we saw there are great examples of size, shape, porosity, distribution going a long way in characterization of complex excipients and complex drug products. But what about chemical composition? What if we have systems that are not binary? What if we cannot really get that contrast from a univariate or from a single parameter, right? So then we need chemical imaging. We need to be able to generate spectra and spatial information at the same time. And then that spectral information can lead us to molecular identification, determination of composition, etc. I really like this painting of William Herschel, who discovered the infrared rays, because it's he used the sunlight. He diffracted it using a prism, and just beyond the red, he placed the thermometer that was blackened and detected the infrared rays for the first time. I really like it because not much has changed in terms of the components we used in chemical imaging in about 220 years. But a lot has changed in the types of components we use. We went from considering mid-infrared or near-infrared, we went from silicon carbide and tungsten halogen lamps to quantum cascade lasers that essentially eliminate the need for an interferometer or a diffraction grating. With Raman spectroscopy, Raman microscopy, we went from helium neon lasers, argon, krypton gas lasers to Nd:YAG lasers, second harmonic generation, and all that. The equipment that would fill up an entire lab to diode lasers that are the size of a fingertip, and holographic gratings, notch filters, the CMOS detectors, and essentially it's pushing us to the limit where we can approach theoretical limits in resolution, in sensitivity, and speed. But also the advancements in mechanical control of stages, where we can obtain this spatial information, the precision that we have, very routinely can be accessible at like 100 nanometers, really rapid, but also the ability to generate large sets of data and process it and at the same time. So not really trying to give a history on this, but really what's available out there right now and in chemical imaging spaces is equipment that can generate diffraction limited chemical maps or images of pharmaceutical products within minutes or hours and process it at the same time. And I'm going to focus on Raman imaging and Raman mapping for a little bit. And where can we apply this? Really, any material that can generate a Raman spectrum. And as long as the spatial features are above the resolution. And OTR at OPQ, we over the past few years we've generated a lot of methods and analytical approaches. We applied them to complex drugs. It's just a couple of examples here. On the left, you see 3 active pharmaceutical ingredient solid dosage form, color coded using a principal component analysis to generate the data. Showing the distribution of each active pharmaceutical ingredient on the tablet surface. And the second one is a crystallization study of a transdermal drug product. We used, in this case, low-frequency Raman. We were able to generate contrast between different crystals, even though this would have looked identical under an optical microscope or an electron microscope. But here we're able to actually identify that these crystals are essentially very different from each other. This is a sunscreen formulation that we just discussed in the electron microscopy image. We're able to visualize different UV filters now, and it's the last one is also a study on a stability study on a transdermal product where we looked at the API crystallization. We could identify what kind of crystal was coming out. I'm going to now move on to a different point, talk about a specific example, which is actually going to lead us to where the limitations are in regards to chemical imaging. So my colleagues talked a lot about this product, and so I'm not going to go into too much detail, but basically micronized API dispersed in the PDMS. So it's a binary system. The Raman spectrum is here. The top is Leavenworth gestural. I'm not going to go and identify every Raman band here, but the point that I'm trying to make here is it's a very busy spectrum. It's in fact the Leavenworth gestural, the API is a very good Raman scatterer. The bands are very strong. It's a very busy spectrum. It has a large Raman cross section. Its inherent ability to Raman scatter light is very strong. Compare that with the PDMS, just the opposite. Very few bands, weak bands, not a very good Raman scatterer. Essentially silicon oxygen or silicon CH3 bands that can be monitored. And just the process, how we go from the collecting a spectrum and doing identification of a material to chemical imaging. The optical image is here from the system. This is zoomed in to show the area of interest. The grid showing where we collect the Raman spectrum. Basically, at each of these points, we collect the Raman spectrum. It's 85 by 50 microns. 1 micron step size, so 4,250 a spectrum, each spectrum containing about 1,000 wave numbers, right? So it's a large data set, has to be processed. In this case, we used an algorithm that's a multivariate curve resolution. We used a least squares approach to an iterative approach to solve it. That's the data. The data set. This is the mathematical problem. Basically, this is the matrix representing contributions, the concentrations, and S is the matrix of pure component spectra. There's a residual matrix. We kind of force the model here. We force it. We ask it to come up with only 2 components because we know it's a two-component system, but we're also assuming that the pure component spectra is within the data set. So the components that we calculated are here. Generally, they're separated. The peaks overlap with the PDMS peaks and the Leavenworth gestural peaks. The images we obtain from using these components, back calculating with the matrix and generating a singular data point for an image. The red indicating the Leavenworth gestural, blue indicating PDMS in the background. So it's a 2-component image. We do a little bit of image processing to improve the quality. But there is one problem that's very easily identifiable. There's some overlapping peaks. We don't really get 2 distinct components. So we worried about the sample preparation because the surface has to be pristine. We worried about when we generate the cross section. We worried about impacts that can arise from how we basically obtain the cross section. We actually used an ultra cryomicrotome for this, which was a very painful process. I do not recommend it for anyone. But we did. There we go. But we did, and we generated not only a cross section but also sections, submicron sections of the sample. And when we applied the same process, what we found was that actually the components that we calculated looked drastically different. And that kind of raised some questions about the validity of applying Raman imaging on a system like this, where the micronized API really approaches the spatial resolution. And that was even reflected in the high-level statistics that we generated with some binarization of the images and thresholding and particle size analysis. So the reason, the short answer is really diffraction limit. You're really essentially approaching the diffraction limit. And even though the Raman response as a function of depth on your sample in a highly confocal system, highly confocal microscope is drastically decreases with the increased distance from the surface of your sample. But essentially, you're really collecting from a cone, collecting data from a cone. And considering that PDMS is translucent, it is not a very good Raman scatterer, whereas the LNG is. So we do have a lot of out of focus contribution that's coming from beneath the surface. So it's not only our analysis here is not biased for larger size particles, but it's also perhaps not representative of the true surface, right? So that's the diffraction limit, and now I'm going to go to in a slightly different direction. Can we beat the diffraction limit, right? So this is possible. You can couple a Raman microscope essentially, basically with an atomic force microscope. And for those that are not familiar with AFM, it's essentially a really sharp tip at the end of a cantilever that is controlled with a feedback loop using a feedback laser and some electronics, and allows topographical analysis of samples, even some mechanical properties at 10 nanometer or sub 10 nanometer resolution. So what happens when you combine it couple it with a Raman microscope with a technique called tip enhanced Raman, you get enhanced Raman signal from the surface, but that is not necessarily representative of a standard Raman spectrum. The last decade has seen a lot of progress in coupling infrared spectroscopy with AFM, and there are three distinct techniques. One is nano-FTIR. That's essentially an FTIR that's coupled with an AFM 10 to 20 nanometer resolution, kind of a complicated system to utilize. There are two other techniques that are very interesting that are using quantum cascade lasers. It's a tunable laser at a specific IR absorption, so you can basically lock it into an absorption band of your material, and so you no longer really need the interferometer or the diffraction grating or anything. And in this case, you no longer even need an optical detector. What's really happening is you focus the IR beam onto your tip and onto your substrate. Two things can happen. One, the IR beam, if it hits an IR absorption band of the material, it can thermally expand. You can detect that thermal expansion using the AFM. Lateral resolution is high, but you need a thick sample, like hundreds of nanometers. And there's another technique called the photo-induced force microscopy, which is what I'm going to focus on. In this case, it's the dipole-dipole interactions between a specialized metal-coated tip and the specimen, and it's sub 10 nanometer resolution, and you can actually obtain a spectrum by using a bimodal AFM. What that really means is the cantilever has two oscillation modes. One you use for topography, and the other one can be used for detecting the photo-induced forces. So without going into too much detail on that, we use this system. We did the same, going back to the same example we had. We did the chemical imaging with Raman before. We went back to the same sample, 50 by 50 microns, 10 nanometer resolution. The topography is on the left, indicating yellow indicating higher points, blue indicating lower points on height. And on the right, we have the photo-induced force image that is from 1654 inverse centimeters. That corresponds with the LNG peak. It's not corresponding with it's unique in terms of comparison with PDMS. So the bright spots indicating the LNG crystal at the surface, dark spots indicating PDMS. And we collect a photo-induced force spectrum that correlates with FTIR. That's really fascinating because it's a completely mechanical detection, and yet it really does correlate with the optical detection. And we can identify the spectrum is a lot more rich in terms of for PDMS when you're looking at it with infrared, and there's a lot of potential to really do complex excipient characterization in this case, but also identification of the sample. So similar to Raman, this can be done in hyperspectrally, collecting a Raman spectrum, an IR spectrum at each point, and processing the data with the principal component analysis here. And we're showing a crystal, a singular crystal here, and the level of detail is really fascinating. Even though it may not be a very it may not be a system that it's just a binary system, but the level of detail we obtain can lead to really very interesting studies regarding the drug release from these types of products. So having established that, the question we really wanted to go after was, what about nanocarriers? Like, can we characterize them? Can we image an API inside a nanoparticle, right? So and it's nanoparticle albumin is a great platform for that, right? Because the albumin it's a highly biocompatible, it's a very successful solid nanoparticle platform that has gained a lot of traction. It's owing to albumin's biocompatibility, biodegradability, non-homogeneity, improve the pharmacokinetics. It's with its chemical structure and conformation, it allows active targeting because it's overexpressed in diseased tissues and cells. That's the hypothesis there. And so much literature is behind it. There are chemical and physical synthesis approaches that are vastly different, can generate different types of albumin nanoparticles, desolvation, emulsification, microfluidic mixing. These are very interesting approaches to it. And there are multiple drug products that are approved, either using paclitaxel as the API or sirolimus. There's, in fact, other drug products that are in development that have more than one API that's inside the albumin nanoparticle. So it's what's also coming is also very interesting. So it's a really good model drug to study. And what's the schematic for the nanoparticle albumin is the albumin shell and paclitaxel in the core, and also smaller fragments of albumin and paclitaxel that are bound together. That's kind of what's proposed. So we wanted to develop a strategy to characterize HSA, a nanoparticle albumin, human serum albumin, nanoparticle albumin, but also nanoparticle paclitaxel that is the actual formulation that's separated via centrifugation to see whether we can chemically image the API and the nanoparticle. So we followed a well-established nanoparticle synthesis route, the disulfide reduction, desolvation, and disulfide interchange, and ended up with nanoparticles that are intermolecularly cross-linked using the disulfide bonds. These nanoparticles were also a similar size compared with the nanoparticle albumin drugs that are in the market. We developed a strategy we used a glass substrate that was carboxylated, very routine EDC NHS chemistry, conjugated using the amide. These are spectra that we collected from our substrate using the photoinduced force microscopy. So the panel on the left, the red spectra is from just the background, the substrate that we had with the NHS, and we see some glass-related peaks and perhaps a carboxyl functional group at 1730. And then we did we conjugated HSA it's in monomeric phase. We obtained the amide2 at 1550, amide1 at 1650. And 1680 is really the broadband, and it's really interesting because the amide1 carries a lot of information that's related to the conformation of the protein alpha helix beta sheet, whether it's aggregated or not. And then the HSA nanoparticle that's in green. And really zooming into this, what was really fascinating was that we could see some differences in the amide1. Some caution has to be exercised. Obviously, we're conjugating the protein onto the sample in dry conditions, so it's very harsh for protein, but it's the same conjugation process, and we do see some differences in the spectrum. And the spectrum, again, correlates with the FTIR that we can obtain. So let's look at some images. The background, not as uniform as we were hoping in terms of topography. Even albumin, we see some, not sure if it's aggregation of the monomer, but it is, there are some patches or areas of albumin that we saw on the surface, but we can trace it with the chemical imaging at 1666, the amide1. And the cross-linked albumin nanoparticle that we synthesized, we also looked at that. This is the topography at the top and the PIF image at the bottom. At 1666, we can again trace the amide1 band, and we can, and this is really fascinating because we can chemically image these particles, and that specific to a particular band in the molecule. So a slightly larger area that's showing the distribution of the particles. This is a very high yield process. And now what's really interesting is we did the centrifugation of the actual product. So let's look at the FTIR spectra quickly. The HSA, again, amide2, amide1, the paclitaxel, The two peaks at 1700 and 1730, these are in crystalline paclitaxel. They don't exist in the, they don't show up in the amorphous paclitaxel. It's also very interesting because now we can not only detect the paclitaxel that's inside the nanoparticle, but also confirm that's non-crystalline paclitaxel, that's amorphous paclitaxel, and the FTIR spectrum from the nanoparticle albumin paclitaxel drug. So we did the separation with the pellet and supernatant, collected the nanoparticles. Not super clear here, but we zoom in and we trace the paclitaxel peak that's at 1645, compare that with the albumin peak. So the supernatant really represents the albumin spectrum, whereas the pellet represents a spectrum that's a combination of paclitaxel and albumin nanoparticle. So it's exciting for us because now we can determine that we can image the API inside the nanocarrier. Looking at some images, some mild aggregation likely happening after centrifugation and resuspension of the particles in the pellet. We're tracing here 1646, which is the API. We see it in the particle in the PIF images. These are chemical images, basically showing that the API is in the particle. We confirm that looking at the spectrum in the previous image. We look at the supernatant. We see a lot of HSA signal in the background. And we do see some particles, but they seem like they're hollow, void, and they don't really have the API here in the supernatant, and that's what's expected when you do the centrifugation. So it's really fascinating for us. But of course, there are a lot of issues with how we prepare these samples. Perhaps we need to do a lot better in conjugation. The conditions that we analyze, the dry conditions, have to be reconsidered. But it's exciting because it's way below the diffraction limit, and it can provide a lot of mechanistic information that can go into guidance development and at the end of the day, determining what needs to be measured and what needs to be and how the drug release is happening and so on and so forth. Just some quick summary of kind of what I wanted to cover. We saw a lot of examples of XRM, SEM, and EDS. It's very high resolution. You can do size, porosity, you can do density. But really, when it comes to the obtaining chemical information, you need Raman. You can do IR, and you can do AFM IR at high resolution. And kind of covered in the last slide, but basically chemical imaging below diffraction limit, visual characterization of complex excipients and APIs, API properties, and elucidation of the chemical composition of the API nanomaterial complexes. And now we can achieve these, and we want to move forward in that direction. With that, I would like to thank the DCDA and my colleagues, Daniel Willett, Jan Yang, and my supervisor, Chang Ying, and Division Director, Jason, my ORA is Vasanti, and our collaborators in an FDA intramural grant, Jiwan and Talak. And with that, thank you for your attention and looking forward to the panel.
Okay, all right. Next up, we have Maxime Merdy. He's a associate director of research and collaboration at Simulation Plus, and he's going to talk to us today about PBPK models and why they're the best.
Thank you, and thank you very much for the invitation. So, which one is it? Yes. So, first disclaimer, I probably understood 10% of everything you have presented over the last 2 days, because my background is really not in formulation at all or excipients. So I've learned a lot. Thank you very much for inviting me. In this presentation, I'm going to talk about PBPK model. And just so we're all on the same base, just want to define briefly what is PBPK. So PBPK is an in silico modeling and simulation tool standing for physiologically based pharmacokinetic model. The idea around PBPK is to combine the system and physiology parameter, that could be the body weight, the BMI, the tissue size, the tissue blood flow, compound specific parameter, the log P, the log D, the permeability, blood to plasma ratio, so all of the physical chemical properties, and formulation parameter that could be the dose, the dosage form, the particle size, the CQAs. And when you combine those three sets of parameters, you can actually start to predict what would be the plasma and tissue concentration for an API. One of the main applications of PPK model is to translate between species and see if the preclinical results that you see for a specific drug product can actually be scaled to human. And that's what I'm going to be talking about in the 2 case studies today. So I'm going to start talking about ocular PPK model. And you can see here on the screen the current structure that we have for the ocular PPK model within GastroPlus. We have an active collaboration with the FDA. So the structure of the model has been evolving a lot over the last few years. And nowadays, so we can describe the ocular, all of the ocular tissues, the most important one, and how the drug is going between those ocular tissues based on passive diffusion, active convection transport, or also just active transporters. There are also other mechanisms we are taking into consideration, such as the T-adrenal rate, for example, and other very important parameters like the transfer between the ocular tissues and the systemic circulation. 2 years ago, actually 3 years ago now, we have entered a new collaboration with the FDA to really focus on how we can use this ocular PPK model to predict human exposure in the ocular tissues once the model has been validated based on preclinical data. We have last year published this first paper focusing on ophthalmic topical solutions, because the idea, we wanted to start very simple and just see if we could predict the absorption of solutions, and then if that could be scaled between rabbits and human. Here, I'm showing on the left so far the results for an API called gatifloxacin. It's an ophthalmic solution administered to rabbits. And we use the first data set that had in green observed cornea PK and observed aqueous humor in 2 studies. We used those data sets to calibrate some of the parameters in the model. And then once the model was calibrated, we predicted 9 additional studies. And those are the 9 plots that you can see at the bottom. So we had a model that was validated and that could predict relatively well the local concentration following topical administration of gatifloxacin in rabbits. What we then did, and going back to the first slide, is we changed all of the physiological parameters between rabbit and human. So we changed the tissue size, we changed the convection flow, we changed the T-adrenal rate, for example, and we kept all of the drug-specific parameters the same, and we just simulated clinical data. And what you see now on the right side of the slide, or the left, depending on how you're looking at, are the prediction of human local data following topical administration of gatifloxacin. We had data collected in patients going through cataract, keratoplasty, and vitrectomy surgery. And we also had conjunctiva data in healthy subjects. There is quite significant interstudy variability, but overall, the model was able to do a really good job at predicting the local concentration in human once it's been validated in rabbits. So we've done 6 or 7 case studies now, and we are very confident that the model can do that for solution. So it was now time to focus on more complex dosage form and see if we could do that as well for suspension. The model that we have initially developed for suspension is considering some key elements, such as the particle size distribution. So we can integrate the drug particle size distribution directly into the model. Once the formulation is administered on the ocular surface, we are going to considering the size-dependent dissolution rate, and that help us to predict what could be the effect of different particle size on the overall absorption. What we're also looking into is the effect of viscosity on the overall absorption process, and by validating the impact of viscosity on the preclinical clearance by affecting the drainage rate. So we have done that work and we have validated these models a while back. And we wanted to use this model for basifloxacin ophthalmic suspension. The drug product name is Basivance. The first model that we used give us reasonable prediction. So you have, starting from left to right, tears, cornea, conjunctiva in yellow, and aqueous humor in red. And if you compare the lines, the simulation with the observed data, they are in a fairly good agreement with the observed data when we look from far. But actually, if we really look now in log scale, and we focus, and you have here on the screen the conjunctiva, cornea, and aqueous humor data, what was interesting to see is the first time points for the first 3 hours were quite well predicted, but the rest of the profile is very not well predicted at all. And we are totally under predicting. And we wanted to understand what is happening because it's a suspension. So we were expecting it's not a function is too complex. So we're expecting to have a reasonable prediction. So we looked at the formulation characteristics and we found that this drug is actually formulated with an excipient called Durosite. And I took this cartoon directly from the website. So Durosite is a polymer that is going to integrate between the ocular surface and the drug. And it is expected that the time contact between the formulation and the ocular surface can go all the way up to 12 hours, whereas for typical suspension, we expect it to be around 15 to 20 minutes maximum. And unfortunately, we didn't have any CQAs for this formulation. So we didn't have any details about the mechanism, but we still wanted to use the data to see what we could do. So what we've done is, instead of having the detailed CQA like you have presented over the last 2 days, we've lumped everything together and we asked the software, can you fit what would be a release profile on the surface of the eye based on the observed data? And this is what you see at the bottom right. The purple line will be the fitted release profile for this suspension. And once we have fitted that, we could describe relatively well the tears, the cornea, the conjunctiva, and the aqueous humor data. So that was quite interesting, but of course we've done fitting, so we would expecting to predict that. Every time we do fitting, we want to have a validation step. So this is what we've done here. We had 3 additional studies in whom we had observed data in cornea, conjunctiva, and aqueous humor, and we simply simulated by adjusting the dose and the study characteristics, but we were able to predict the data relatively well. One disclaimer is when we talk about rabbit ocular studies, there is a very significant interstudy variability that we cannot account yet. So that's why for some of the studies, it's not perfect prediction, but as a whole, we have a really good prediction of the observed data. So we are quite happy with those simulations as well. Then we wanted to see, can we go to human using the same release profile? Oops. And this is what we got. So here you have the human prediction in tears on the left, aqueous humor in the middle, and conjunctiva on the right, coming from 3 different studies. And similar to what we've done to solutions, we've just changed the physiological parameters. We kept all of the drug-specific parameters the same, and the formulation-specific, aka the release profile, the same. And by doing so, we were able to have a really good prediction of human PK. So what was interesting for us is that at this stage, although we don't integrate the CQ in the model, and that's the main limitation, it seems that what is happening in rabbits is probably also occurring in human. And now what we are hoping is, can we actually go study in vitro what is actually happening for this drug product? What is the interaction between the drug and the Durosite? What is the impact of this on the surface of the eye? How can we improve the model to mechanistically describe the interaction between the excipient and the surface of the eye, and then the release of the drug? Refine the model, revalidate on rabbit, and repredict the human. But we are quite confident that once the model will evolve, we'll be able to do that. The other case studies I wanted to talk about is for long-acting injectables. And a couple of years ago, we have entered a collaboration with Dr. Burgess at UConn, and we started to improve the model. So the long-acting injectable PPK models that we have in the software will consider the drug depot or the drug depot volume, but that volume may change based on time, especially due to inflammation. We are considering the particle size distribution, we are considering the drug dissolution and the release rate, the partitioning and the absorption between the sub-Q and the plasma, and then the systemic distribution. What we started with is actually a set of data that was already produced by Dr. Burgess and his labs, and our labs, looking at one reference listed drug for MPA suspension, LAI suspension, and 4 formulation variants. And we also had IV data for this drug product in rabbits. The first thing we had to do is to make sure the IV data were well predicted. Because if you don't do that, and you want to model plasma for other drug products, if you don't fit well the IV data, you won't be able to know whether the misprediction is coming from the absorption or from the distribution. So here we have the IV data, and we had a really good prediction of the observed data. So we are quite confident that if we see a misfit, it can only come from what is happening in the depot site. We then start to look at the OLD, and that was the initial simulations we got using all of the default parameters in GastroPlus. Interestingly, the AUC was actually not too bad, but when you look at the Cmax and the shape of the profile, it's not a good prediction at all. The first thing we've been doing is to increase the diffusion layer thickness to account for the somewhat static environment of the LAI, of the surrounding environment of the LAI. And when we did that, we're able to have a better shape of the profile, but it was still not perfect. We then try to scale the measured in vitro particle size distribution to account for what we think may happen is particle aggregation. So we started to scale that and see whether we could improve the prediction, and we had slightly improved, but you can see the first phase of the profile is still like mispredicted. So finally, what we have integrated is the effect of inflammation and how there is a transient increase in the depot volume for the first 2-3 days following the administration. And once we are considering all of those different phenomena, both on the formulation aspects, but also on the physical response, we're able to have a reasonable description of the observed data in rabbit once again. We kept that same strategy, and we use it to predict the 4 additional formulation variants and see what the results could be. And if we look as a whole, we have a fairly good prediction of the observed data for the 4 additional formulation variants. If I look here, I'm not going to go into the details, but the Cmax and AUC observed and simulated, they're all within 1.5 or 2-fold, which are really good criteria when we talk about PPK validation. So we had this model, and we wanted to see, can we predict human? Can we go a step further? So that's what we've done. And if you look at the blue curve first, that was the prediction we have obtained when we used all of the parameters we had fitted based on the rabbit study. And once again, if you look at the table on the right, you can see the prediction errors are less than 2-fold, which for first-in-human application will be totally accurate. But we wanted to see, like, can we actually improve the prediction? Can we go a step further? And when we changed the physical response, and especially the inflammation parameter, because the rabbit seems to have like quite a lot of inflammation compared to human, we were now able to have a really good description of the PK profile, and that's the black line versus the observed data. So to conclude and to summarize, if I go back to the ocular model, the ocular PPK model, it presents an interesting approach to support regulatory assessment of complex generic drug products, knowing there is still some room for improvement. One of the challenges that we see, especially for all of the non-solution drug products, is we need to have a better identification of formulation CQAs and what could be excipient impact on the overall absorption process. So that is the next steps that we are going to look into in the near future. For LAI, one of the very specific characteristics is we can connect the formulation characteristic as well as the local response following the injection. And it can really help us to differentiate what's the impact of one and another and what is the cross impact of those effects. So same, we're going to do more work in that space to make sure that we can have a very refined model that can really support development. But on and all, if we look at both case studies, And it's really showcasing that one of the unique advantage of PPK model is due to their mechanistic structure that is linking CQAs and physiology. We can actually predict what is the effect of both CQA changes, but also the physiology response to those, and how we can go from in vitro to animal, in vitro to human, and animal to human. And we can really connect the dots of all of the work that you've been doing, going from like really the in vitro and the formulation characteristics, and start to predict what could be the impact in human, and therefore informing regulatory decision for generic drug products. With that, I'd like to acknowledge all of my former colleagues at the FDA and the people I'm working on a monthly basis, but also all of the scientists working at Simulation Plus, and especially Dr. Daniela Silva, who's been doing all of the case studies for LAI. I'd like to thank Dr. Burgess from UConn, for whom we have this very nice collaboration, and also all of the collaborators that we have globally. So thank you very much, and happy to take any questions during the next session.
Okay, I'd like to invite all the speakers and panelists back to the stage. Whoa. Okay, so besides our speakers, we also have two experts on the stage today with us. We have Brenda Pillari. I'm hoping I'm saying that right correctly. Head of Regulatory Affairs at Viatris. I don't know, Vitris?
Former Mylan.
Former Mylar?
Mylan.
Mylan, my bad. I'm bad with names. And we also have Khondoker Alam, who is down there at the end. I'm trying to find your exact affiliation at the FDA. It's okay. I'll find it later. Oh yeah, senior staff fellow in OGD, of course. So yeah, does anybody have anything to kick us off with? Oh, it looks like, yeah, Steve back there.
Hello, thank you very much for the excellent presentations. I have a question regarding the imaging of the in situ forming implants with the Atrigel. And first of all, I think if this can go forward, I think it's really a terrific thing. Whenever and we put dyes in, for example, to measure pH and so on, and one of the things that we're very always concerned about is whether or not you're imposing any change to your delivery system when you add such a agent like this imaging agent that you cleverly put in there. And so I may have missed it, and I saw lots of controls in your slide, but did you show the release of the leuprolide without the imaging agent versus the release with the imaging agent?
Yes, I did.
You did. I'm sorry, I missed it. So they were the same.
Yeah, great point. That's also the first concern we had. If we use any of surrogate, we want to make sure it doesn't influence the original. So we compared without any drug. We compared with leuprolide acetate only and ihexyl only, and then mixture. Basically, when you see wherever there is leuprolide acetate, ihexyl will just have the same performance as leuprolide acetate. So
yeah, I saw the comparison of the dye and the leuprolide in the dye, but I don't know that I saw the dye and
they're very consistent, like the same curve.
So the other thing is, have you checked to see whether the dye has any plasticization of the polymer?
So as any
right, does the dye interact with the polymer?
So if it does, and then you don't see the difference between the die by itself or
Right.
contrast agent by itself versus the mixture. Basically, I don't think.
I see. Oh, so,
Yeah.
okay. Well, anyway, I just, I'll review your slides again when I was maybe sleeping, but I missed that one point. Thank you.
Thank you.
Okay, it's working. So I have a question. So today we saw a lot of new approaches to characterize particles, particle size, so new instruments and so on. So I remember that in 2007, FDA provided an option critical path opportunity for locally acting nasal spray suspensions. If you are able to validate and develop Raman microscopy method that you can waive phase 3 studies. So at that time we tried, we saw that the instruments were still not that advanced that could give us reliable results. So at that time we concluded that it was not possible, but we've seen in the last years, in the last batches of product-specific guidances that were releases, that this option now it's clearly part of the guidance. So obviously also instruments advanced to that level that this analysis is possible. How do you see these techniques that were presented today? What is the potential that these methods can be validated and used to generate amount of data that is required, for example, I don't know, for the ophthalmic products where the particle size can be expected below micron range, meaning that we need to be able to do this in a reliable way and with the correct technique. How do you see this? Can we expect this soon, or what is your opinion on this?
That's a very good question. I think it's very difficult to predict when it's going to become, when the technology is going to mature, because it's going to need a lot of user experience. Like you said, when the Raman microscopes, confocal Raman microscopes came, it's been a long time. But it took a while for actually someone to publish a paper that really showed all this 1 micron resolution. It wasn't really practically what was happening, right? So there's going to be hype. There's going to be, the up and then the down, the maturization of the technology, as well as the user experience with it. But it is out there. The validation is another question, I think. Especially, I think you mentioned orally inhaled drug products, right? Did you mention SCP with orally inhaled or oral inhalation products? Nasal, okay. Okay, right. So the I think now like the MDRS is almost very routinely used for that, right? But there are other techniques that they can come into that space and fill a lot of the scientific gaps, but also analytical gaps that remain, because they do remain. It's going to depend a lot on the user side, I think. Because the technology is there, the Raman microscopes were miniaturized. They were developed 20 years ago, right? So, but it took a lot of time for people to actually use them to see what the drawbacks are, and that's it has to come through that, I think.
So from an industry perspective, the key towards actually embracing, these new technologies, which are awesome. Thank you for all the presentations. For characterizing the complex products during product development and assessment is developing the acceptance criteria for assessing the data. And we're going to talk more about that in the breakout session. So that's great. But also having the agency and standards organizations embracing the technologies. I think from an industry perspective, we're all also going to have to weigh the cost of the testing, validation, and any difficulties within the analyses. So I'm not sure how difficult these technologies are to perform the testing, but that's another question mark for industry to explore. But certainly information sharing, publications of this type of these types of technologies, FDA and USP engagements, global information sharing. I have a note here that potentially a repository of information for these new technologies on the CRCG website might be beneficial as well.
Okay, I got one to follow up here, and maybe kind of following on to the regulatory thing. Do you guys kind of like, what are you envisioning as maybe some potential issues or regulatory challenges that you might see to applications that are using model-based kind of approaches to bioequivalence? Maybe, I don't know if Alan wants to talk to that, or if any of you guys have kind of some prediction on that.
Yeah, very nice question. So we are getting a lot of P&D applications recent time where applicants want to use the model-based approach, and we often see the good, reasonably good model that is developed well and validated well. But the thing that is missing from their application is that how the model will be utilized in the for their proposed approach. For example, for the bioequivalence approach. So model-based approach is not about only limited to model development or validation. We need to give some answer that how the model will be used to detect the potential formulation difference, and how we are going to choose the subject numbers, and what is the basis of that selection, and are you going to incorporate any inter or inter-subject variability? These are very tricky to characterize, I understand, but some kind of reasonable assumption on the inter-subject variability is always helpful. And there are other aspects that they need to also consider, that the risk assessment like type 1 and type 2 error, that should also come through their application package, that this is the way we want to use our model for the for supporting their alternative bioequivalence approach.
Anybody else want to attack a little bit on that? Okay. So I'm also wondering, maybe Shawn, as far as like AI assisted image analysis, as far as like when you're trying to like train some of these models to do, like, say, pore detection or density discrimination, what consideration are you putting towards having enough, say, like RLD product lots for training your sets, I guess?
Yeah, another great question, Billy. AI brought a lot of buzz, a lot of excitement on one side, but also probably similar amount of questions, right? Is a ChatGPT answer valid? Is there a critical quality attribute we can assess a ChatGPT answer is valid or not? Right. So this is getting into really new territory while the technology is advancing, and along the way taking many years of human knowledge. I think a few months ago, we had this great discussion in another workshop regarding the AI regulatory pathway about the AI. So even though ChatGPT is becoming famous overnight, but our collective scientific effort of getting to today's ChatGPT success happened over the past 3 decades, right? That went through a very consistent digitization, digital transformation of our human knowledge, our human information into the digital space, making them accessible. Right. So that's the publicitization part, the institutionalization, right? Making them broadly accessible and making them consistent. Things like Wikipedia, many years of moderation, a lot of voluntary efforts. cleaning the data, curate the data. So it takes time, right? Coming back to image analysis, so using our experience, so we are collecting a few thousand images on a daily basis on various kinds of products. Right? So AI and AI-assisted automation is first and foremost a necessity. Right? We simply can't afford going through all the images, identifying each particles manually. That's just not possible. Right? So with that said, between AI can do ChatGPT or just throw an RLD and tell me the pathway to develop a generic. Before we get there, that might be the dreamland. Right? AI can also do a lot of things in prior. Right? So image processing, vision applications, these were widely applied outside pharmaceutical development industry. Right? We have actually a lot of existing knowledge we can tap into. And the validation is also a good, putting us into a very good groundwork. Now, why we adopt this for pharmaceutical applications, that's still not a necessarily easy job. Right. So directly to your question, there are actually several approaches in the validation of AI results, just like any actually analytical results. Right. So we have this cross blindfold design. Right? We have multiple users. That could be actually some of our internal users, some of the external users. We actually have a very open digital transformation framework, welcoming each and every our collaborator coming here and train the AI alongside with us or independently. It's the same image, right? I can train it. My colleague Andrew can train it, or one of you can train it, and we can see how the results look like. So this is one important cross validation. And then back to the earlier question regarding the particle sizing, right? So we have the historical measurement data, and then we have the different parallel measurement data, laser diffractions, wet or dry. So all of these cross validations is also another really important anchor point for our AI model. So I think in short, while it does require some kind of mentality change and sometimes a courageous step, right, toward embracing this newer technology, but at the same time, cautiously optimistic, right? We need to assess all our scientific discretion, use all the scientific steps to assess each and every new technology embracement that we are doing.
Nice. Oh, yeah, go ahead.
I have a question for Maxime. So you showed a very nice case, right? The promising extrapolation from rabbit eye data to human. So the first question is, besides the case you just presented, do you have other cases you have seen this promising extrapolation?
So for solutions, we found like around 10 to 12 case studies in literature where we could do those extrapolation, and it's been working for all of them. So for solutions, yes, there's a bunch. For suspensions, there are only 2 case studies with published data, dexamethasone and becifloxacin. And that's honestly the main challenge is there are very limited clinical data available to validate extrapolation. So we can find PK data in rabbits for most of the suspension, but there is almost nothing in human. So we've made the extrapolation for dexamethasone is working quite well, but that's like one of the simple suspension. So using the default suspension parameters, it worked quite well. For becifloxacin, we had to make the trick as I've presented. But unfortunately, we're going to be limited to those 2 case studies until more data are generated, or we can find more data maybe in some collaborators' databases or something.
Yeah, I mean, the reason I'm asking is I think PBPK model has been around for years, and applications especially in the new drug side, right, to better understand the in vivo clinical, purely clinical post administration or sometimes post absorption. But for generic space, the difference is you have to incorporate the considerations of critical quality attributes of the formulation, which is prior, right, to what conventionally has been done. So the validation of the model can be quite a target, right? A challenge, I would say. But then if we have enough understanding or evidence to really show how animal data may play a role, although it's something new, but I guess that's something, I was just curious to see how much progress has been made and, what's your view on that?
No, yeah, we're just limited by the data. So for suspension, there's only one case study where there are published data. For gels, there are 0. For emulsions, there are 1 case studies where we have also like published clinical data with local concentration. So that's going to be our main limitation is because we are a software company, so we don't generate data. So we only use like publicly available PK. There are not that many.
I can add a little more on that aspects. Yeah. So I see that animal studies are useful in model development purposes. We know that if we inject the long-acting injectable suspensions, there is a form of depot and collagen fibrous envelopes and how they are changing over the time. So we can characterize those things with the animal, with the imaging technology that we have seen in this session. So that can be used to inform the model, but when it comes to the regulatory decision or to support the drug approval, I think that animal model or animal data, PK data, shouldn't be used that purpose. We should use the human PK data only. That is my thinking. Yeah, thank you.
Yeah, I agree. I mean, from regulatory perspective, but personally, if you consider generic space, right, it's a comparative testing. So, if we think we are testing things through in vitro, subject them into the same method, even we recognize from method to method, there could be variation, but as long as both are subjected to the same condition, then that variability can become like normalized, right? So if you take that approach personally, this is pure personally, I feel like the mindset for how you consider animal for generic could be different.
Okay, I have some more technical questions from the internet. I guess one of the questions here is for Dr. Liu. What differences were you seeing between implant morphology in vivo and in vitro, and were you able to distinguish any kind of density difference at the interfaces of those implants?
Right, so we see a bulk trend, similarity, consistency between in vitro and in vivo. For example, that iHexo only, you can see a core shell structure the same way in vivo as you see that very bright core in vitro. And this with nucleic acetate, and you see more dispersed, well dispersed. And so that morphology wise is similar, but they will have different trends, and then we are able to see more of a drug accumulation location differences. So, but I don't have very quantitative comparison. I don't want to make any conclusion at this point. It's more of a visual, and later we will correlate that into drug release because we have another way to make it very precise in real-time monitoring your drug release, and then in combination with the image-based analysis in terms of the size and shape and volume like that to make a conclusion. So at this point, I would say the trend is similar.
Mm-hmm.
Yeah.
Okay. No more. Okay. Next one's for Shawn. Do you think that the difference between the porosities were that the interior pores were accessible for the your imaging, but not intrudable for the mercury? So you had that second I guess they're referring to that flat region in the red curve.
Yeah, so that's a really great question, and I guess maybe part of the reason I got 4,000 followers, either maybe half of them don't believe at all what I said, but just want to check it out. So that's a micro porosity system we are dealing with. One other important consideration in comparison with when these more conventional microstructure porosity particle technique were developed many years ago, right? Mercury porosity being a good example. They were designed to study a rigid material, like geological samples, ceramic samples. They are becoming industrial standards, are very carefully validated. But in the pharmaceutical industry, we're dealing soft polymer material, right? So if you remember that curve, I have the 3 sections, right? The first section, what we call the intermicrosphere porosity, those are relatively well established. So Mercury can measure them accurately, but they're not interesting to us, right? Now, the second scale, what we call meso porosity. So that's the intramicrosphere porosity, a few 100 nanometer to a few micron. Those are the porosity of interests. However, as the mercury or some external fluid intruding into that pore space, a few 100 nanometer to a few micron, it requires pressure, right? And that pressure is exercising equal pressure force on the pore space as well as the polymer matrix hosting them. So effectively, what we believe is at that pressure range, mercury pressure is actually closing all the porosity of interest. That's why we actually have a flat. We do not have any porosity measurement. And then later on, as the pressure continues to increase, we see some additional pressure reading. So my polymer physicist colleague Andrew and I are having an active debate where exactly that porosity are coming from. But my personal opinion, we are actually looking at mercury molecules being penetrated into intrapolymer molecules, the long chains. Some of the intramolecule space there. And the reason is we are looking at a few nanometer to 10 nanometer. So just putting in context, this corresponds to about 10 to 20,000 atmosphere pressure. Right. So this is where we are looking at a very different domain. And in those high pressure, even ceramic samples and rock samples are subjected to deformation. And think about our soft polymer chain.
Oh, yeah, we got one more out there.
So I have a follow-up question to you, Shawn, on this. First, it was very rightly mentioned that for soft materials, right? So MIP was not made for that. And we have also, many people might have experienced the lipids and other microparticles. You very nicely showed that in the mesoscopic range, basically you nullify that. But the molecular range, just you just explained there, you still see the classical BET signal, right? The 20 nanometer to 100 nanometer. So my point is, because MIP has a legacy, there's theories. So if you still could discriminate that region, would that be discriminated as well? Because I mean, the molecules, mercury needs to go to invade there. So would that be also indicative? So that's okay. Not for more question from the MIP side. Maybe you don't have answer.
That's also, I think, a valid consideration. And this actually gets to our earlier discussion regarding potential Q3 properties of the polymer matrix itself, right? So we can argue, those parts can potentially be still material relevant and maybe also therapeutic relevant, right? So the other counter consideration is if a measurement technique is penetrating into the molecular chain, do we consider actually the material is being damaged? Is it actually still representative to the actual measurement we care about?
I mean, in polymers, like we know in tableting, right? Because it's pressure, but then you have more plastic deformation effect probably there. So you have that viscoelastic transition. Like you said, my question is if we can relate that with the precedent measure structure, for example, then MIP is fairly easy, direct technique. You are really measuring direct, basically observation, the intrusion volume, right? So that was like, if that region is resolving, so it's a model free, basically.
Yeah, I agree. I think that last bit of porosity or nanoporosity behavior requires a bit more research and investigation. But I do want to also mention actually a fairly thorough cross-validation. Now switch gear into the biopharmaceutics. That's also underway in comparing this imaging-based, AI-based technique with autosorption based technique. So there's also a very interesting comparison coming up, most likely in the IFPAC talk in a few months. So I think, long story short, all these conventional physical techniques, right? They have their applicability. They have a lot of good in them, but at the same time, they have limitations. And some of those limitations can be well compensated by these later, in our case, imaging-based, but some later analytical techniques.
Thanks, Shawn. So I have one more question to Maxime's presentation, which was very interesting. So especially for the long-acting case, so it turned out that inflammation is a major contributor, right, in your wedding. And we also discussed a little bit offline yesterday. So that's an area where we still need a lot of clinical data or attention at least. So this is, did you went there from iterative fitting, or is there at least for a rabbit report on the increase in volume? Because inflammation had other cascades of effect, right?
Yeah, so the way we approach the question is we found a study that actually measured the inflammation in rabbit for another drug product. And from that, we could derive the kind of the inflammation rate, so like from going up and down. And we have fitted those parameters based on that study and used them in our models. So we actually have this kind of bell curve that is reproducing the inflammation in the model. And that is then connecting to the total depot volume. So the volume is going to be increased based on the inflammation, and then affects the overall dissolution or transfer rate between the depot site and the systemic circulation. So that's how we approach the question.
Because my experience is I've worked in different systems like nanocrystalline long-acting suspensions. And in those cases, 2013 or sometime, we are done in paliperidone palmitate. And these were simply taken by macrophages, and it's temporal effect, basically. So that's a different kind of, so your dissolution when crystals are happening inside macrophage.
Yes, so the palmitate is a different one. We have, okay, we worked on this one.
It's published one, I mean.
And for this one, what we actually implement in the software is actually the creation of the immune cell layer that is surrounding the depot, and how is that affecting the overall transfer from the, so it doesn't affect the dissolution, it's really the transfer from the depot site into the surrounding environment and then the systemic. So we also have that case studies on. It's going to be published next year, I think.
Thank you.
Okay. I guess I have one more online question here quick. So it seems like the online chemical mapping of nanomaterials for using the AFM is really powerful. You discussed basically functionalizing a glass surface for albumin, but a lot of these other products, including like a nanocrystalline suspension or say emulsions, or what are some of the practical kind of sample preparation challenges or kind of things that you could apply this technique to that aren't necessarily powder-based?
There are more innovative substrates that are now available, commercially available, that three-dimensional substrates that might be like a hydrogel, that may be more conducive to aggregation and things like that. And that's one approach that can be taken. If the formulation is complex and has a lot of different fractions, it can be used in tandem with better separation techniques. I think that's like SEC for collecting fractions and going to AFM. It's really with the substrates, I think it's really a matter of seeing what's available out there, and we're working with the vendors that develop the substrates.
Nice. Okay, so with that, I'm gonna pass this off, I think, back to James. Can we all please thank our panel? Thanks, guys.